So, just a few more definitions. Hypothesis, again, explanation for an observation that can be tested by additional observations and experimentation. Prediction is the outcome expected if your hypothesis is correct. And an observation is the act of noting or perceiving your objects or events using your 5 senses. Upon arriving home from school, Ramon finds that his TV will not turn on. He finds that the TV is plugged in, so he guesses that the batteries in the remote control must need replacing. He replaces the batteries and the TV turns on. In this very simple example, Ramon has just applied some elements of the scientific method. When Ramon assumed the remote control batteries needed replacing, what was he doing? A, making an observation. B, forming a hypothesis. C, conducting an experiment to test a hypothesis. Or D, using a control group. Okay, so the first thing that happened was Ramon found that his TV will not turn on. What is that? A, B, C, or D? That should be A. He observed that his TV will not turn on. When he found that the TV was plugged in, he guessed the batteries must need replacing. So that was him forming a hypothesis. When he conducted the experiment to test the hypothesis, that was when he replaced the batteries and the TV turned on. So C, the conducting the experiment to test the hypothesis is when he replaced the batteries. Okay, he did not have a control group, so it cannot be D. So the answer was when he assumed the remote control batteries needed replacing, that was forming the hypothesis. When he actually replaced the batteries, that was conducting the experiment. So when he assumed, that was his guess, that was when he formed the hypothesis. Elements of a good experiment. This is what makes a good experiment. So the opposite of these things are going to make a bad experiment or an invalid experiment. So good is also known as valid in science. So increased population size. The more the better. So the more subjects, the more participants you have, the more valid your results will be. It helps eliminate random errors. Like we talked about earlier, if 1 out of 10 people get COVID-19 versus 1 out of 100, that's 10% versus 1% of your results being messed up. So it will be better to have more people. Control group, that's a predictable group. You know what to expect. So it receives no experimental treatment, and it's used to compare with a variable group. Another name for the variable group is the experimental group or the tested group. So it's used to compare what's normal with what's new, the experimental group. So remember, the experimental group gets something extra. It gets something extra. So this is the group, that extra thing is called the independent variable. So it's a group containing your independent variable, and when compared to the control group, it is compared to the control group. And so it should be identical to the control group when it's compared to it with only one thing different, your independent variable. So it'll have one extra thing added to it. A researcher hypothesizes that a small amount of caffeine has a positive effect on memory. 50 people were given a small amount of caffeine before taking a memorization test. The average test score was 85%. Which of the following explains why the above experiment is not a valid test of the researcher's hypothesis? So your hypothesis is that a small amount of caffeine has a positive effect on memory. So is it A? So what makes this not valid? The sample size of 50 was too small. B, caffeine has been shown not to affect memory. C, the average score was not high enough. Or D, there was no control group. So the sample size of 50 being too small, that doesn't necessarily make it invalid. Caffeine has been shown not to affect memory. We don't have any proof of that. Caffeine actually does show to increase your mental capacity or your mental quickness. average score was not high enough, we don't know what the average score was normally for these people. The reason why we don't know what the average score was normally for these people is because there was no control group. And remember, we always need a control group. So that is your best answer. The result of a scientific experiment is probably valid if A, most people believe the result is correct. B, repetitions give consistent data. C, it is based on a hypothesis. D, it is done by a world-famous scientist. So, what makes it probably valid? Most people believe the result is correct. Let me tell you something. When the coronavirus first began, Corona beer sales dropped by 8% because people thought drinking Corona beer caused the coronavirus. So obviously we know that beer doesn't cause viruses. So just because most people or some people believe the result is correct doesn't mean it's correct. So that's definitely not going to make it valid. If repetitions give consistent data, yes, repetitions giving the same data over and over will make an experiment valid. If it is based on a hypothesis, that doesn't mean it's valid, because I can make any hypothesis. That doesn't make it valid. If it is done by a world-famous scientist, well, I hate to tell you, but even world-famous scientists might not get it right every time. Okay? It usually takes lots and lots of trials to get things right, so that does not make it valid. Your best answer is B. In a controlled experiment, 25 laboratory mice of similar size and age are divided into 5 groups with 5 mice each. 4 of the groups are given doses of an experimental drug. 1 group is given the placebo. What is the control group in this experiment? The scientist, the drug, the placebo, or total number of mice? If you remember, I told you the control group is always the placebo. Remember, the drug is going to be your experimental group. The scientist is just a scientist. And the total number of mice, that's not anything. The difference between an experimental group and a control group is that A, the experimental group has a known outcome. B, the experimental group contains the variable being tested. C, the control group contains no variables being tested. D, the control group is needed only when testing more than one variable. So let's automatically eliminate 2 choices. Which 2? Should be A, we don't have a known outcome for anything. We have a guess. So A is incorrect. And D, the control group is always needed, not only when testing more than one variable. Now, I want you to really pay attention to this part. This is difficult for people. You're looking at the difference between the 2 groups. What have I said over and over is the difference between the 2 groups? Okay, another way to think of it. Which statement can tell me what is different between the 2 groups? Is it the experimental group contains the variable being tested? Or the control group contains no variables being tested? These are both true statements, but which one actually tells me the difference between the 2? The correct answer is B. The experimental group contains the independent variable being tested. Remember, the experimental group contains what's different between the 2 groups, and that's why I've said that over and over. Now, the control group does contain no variables being tested. This is a true statement, but it does not give me the difference. It doesn't tell me what the other group has. All right, so another element of a good experiment is having an independent and dependent variable. It's not an experiment if you don't measure anything. It's not an experiment if you don't have a difference between your groups. So remember, your independent variable is the only difference between the 2 groups, the control group and experimental group, also known as the variable group. And usually your independent variable is what you are testing the effect of in your experiment. So that's what changes, what's added, what's extra. Okay, I'm changing the amount of fertilizer, I'm changing the amount of oregano. And then my dependent variable is my result, what I'm measuring. Your results are measured, right? Okay, so this is usually something you're measuring, and it's going to be caused by the effect of the independent variable. So my independent variable, my fertilizer, is going to cause plant growth. So my fertilizer is going to cause the plants to grow. My independent variable is going to cause my dependent variable. My oregano is going to cause the taste to be better in my lasagna. Does that make sense? If not, please ask me a question. You can do that on the discussion board. The control group in a correctly designed experiment is most likely A, a smaller size than the experimental group, B, the same size as the experimental group but receives no experimental treatment, C, the same size as the experimental group and receives only part of the experimental treatment, or D, a larger size than the experimental group. So the control group, should it be smaller than the experimental group? No. Should it be larger? No. I think we can all agree that it should be the same size. So should it be the same size but receives no experimental treatment, or the same size and receives part of the experimental treatment? Remember, we've said over and over the control group is your normal group, so it should not receive any experimental treatment. So B is your correct answer. So again, independent variables, what can change? Dependent variables, what's measured. So what can change is the amount of fertilizer I put on something, and the amount of oregano I put on my lasagna. What I'm going to measure is the growth or the height of the plant, so I can compare my control group with my experimental group to see which one got the most growth, or that's not correct grammar, but just go with it. I'm tired. So the dependent variable could also be the taste. What tastes better in my lasagna? So reviewing here, steps of the scientific method, you're going to observe, collect all your observations on your problem, ask a question of how you can fix this problem and do your research, form your hypothesis, and make your predictions on which way your hypothesis will go. Then do your experiment, collect your data, and draw your conclusions to reject or accept your hypothesis. To do an effective investigation of the damage caused by acid rain in a certain region, which is caused by air pollution, all of the following methods should be used except. So 3 will be good, 1 will be bad. We are looking for what would be bad. A, forming a hypothesis as to possible causes of acid rain in a region. Is that good? Yes, that would be good. We're looking for bad, so we're going to cross that one out. B, conducting pH tests on bodies of water within the region and comparing acidity to normal rainwater. That would be good. Looking at the pH to tell the acidity of the lakes and rivers, to compare that to normal rainwater to see if the lakes are collecting more acid because of acid rain. That would be good. All right, so you should basically be between C and D. C is investigating weather patterns to locate winds that might sweep pollutants into the region. Or D, keeping rainfall and temperature readings throughout the year to measure any increases in thermal heating. So this is a commonly missed question. So you have to look at what you're actually investigating and measuring, okay? So in this case, you're measuring thermal heating, which really doesn't have anything to do with acid rain. They put rainfall here to trick you. Okay, so what they're looking at is increasing in thermal heating. That's not going to help you to do an investigation on the damage caused by acid rain. What will help you looking at damage of acid rain is to see how the winds are moving pollutants into a region. Okay, so let me show you what I'm talking about. So if I'm not a good artist. Let's pretend that's the United States. If the winds, weather tends to have certain patterns. If the weather patterns tend to go this way and you see the winds going this way, then that's going to help sweep acid rain into a region. It's going to move the weather into that direction. The winds move the weather into that direction. One example of this is a heavily populated city. One of the most polluted cities in the world would be Mexico City in Mexico. And so acid rain is very prevalent because right outside, because the weather patterns move the pollution, the emissions from vehicles, from older cars. It goes into the atmosphere and it causes the acid rain, and the wind moves it into the areas surrounding Mexico City. So the worst acid rain will be outside of Mexico City because of that, because of the weather moving the pollutants. Which of the following is the proper sequence of events for the scientific method? A, experiment, then hypothesis, then conclusion. B, theory, then hypothesis, then experiment. C, hypothesis, then experiment, then theory. Or D, theory, then conclusion, then hypothesis. Well, hopefully you realize theory is last, always. So we're going to eliminate those two. And let's look at A. Would you have your experiment before you made a hypothesis? No, you wouldn't. These should be swapped, which would give you C. You formulate a hypothesis first, then you experiment. After you do repeated experiments, then you can get a theory. Which of these is true of a proper scientific experiment being conducted under reasonably similar conditions? A, it should provide identical results. B, anyone should be able to repeat it. C, it should disprove a hypothesis. Or D, anyone should be able to influence the results. Looking at A, should it always make identical results? Not necessarily. Just because you're conducting it under reasonably similar conditions doesn't mean you're going to get identical results. Should anyone be able to repeat that experiment? Yes, that is very important. You have to be able to repeat the experiment if it's a proper experiment. You've got to give good directions so that anyone can repeat it. If it's not repeatable, it's not a good experiment. Should it disprove a hypothesis? No, sometimes it's going to support a hypothesis. It could go either way. So that doesn't mean it's a proper scientific experiment if it disproves a hypothesis. Anyone should be able to influence. Influencing results is bias. Bias is bad. Okay, a proper experiment should not have bias. So, B, anyone should be able to repeat it because you gave good directions. Graphs. Graphs are excellent visual summarizations of data, and it can show you what your data is, your conclusion. It can show you which way your data is going. Like I said, it's much easier to look at a graph or even a table, to be honest, but it's much easier to look at a graph than paragraphs and paragraphs and paragraphs of written paragraph data, words. Okay, graphs can show you very quickly what area is larger or higher. And they are typically better than a data table, just looking at a bunch of numbers. They're constructed to help analyze your results and predict trends. If your graph is, if here's your graph and you're plotting it and it's going this way, what's it probably going to do? It's probably going to keep going that way, right? So you can predict future trends. So you can analyze your results and predict trends, future trends. So anyway, three main types of graphs: line, bar, and pie charts. There are two axes. So remember, the x-axis is the left to right axis. So that's my x-axis. It goes horizontal, and that is where your independent is plotted, your independent variable. Your y-axis is your vertical or up and down axis, and that's where your dependent variable is plotted. So here again, your y-axis, dependent, x-axis, independent. So a line graph is going to show continuous data that never stops. If it's continuous, it never stops. I mean, unless your experiment stops. Good example of this is the height of plants. Does a plant stop growing? Not unless it dies. So the height of a plant is going to continually go up and up and up and up. Another example is the amount of fertilizer you put on a plant. If I put 10 grams of fertilizer on the plant once a week, that 10 grams I put on it last week didn't just go away. You just have to add to it because the nutrients get depleted. So the 10 grams is still there. It just moves locations to the inside of the plant, if that makes sense. Time is continuous. Time doesn't stop. We can't stop time. So if you're measuring time, then a line graph would be good to use. Bar graphs are great to show categorized or counted data. One example is the months in a year. You can look at the, how many people did something during October. Looking at brands of things, looking at the number of things. Bar graphs really are good, just multi-purpose graphs. When in doubt, I would say use a bar graph. So bar graphs, one example would be if I can group people by 9th grade, 10th grade, 11th grade, 12th grade. I could say, how many people Miss Bell teaches? Well, this semester, I'm going to have a high number of 9th graders and 10th graders and very few 11th graders and no 12th graders. Next semester, I will have probably no 9th graders because the classes I'm teaching are changing. And I will have probably a few 10th graders, but a lot of 11th graders, and maybe a lot of seniors. Okay, so I could use a bar graph to look at that. A pie chart is going to be used for percents. That doesn't mean the others can't show percents, but usually this is a percent of the whole 100%. Okay, so if something can add up to equal 100%, then a pie chart would be good. I could also use a pie chart to look at the distribution of 9th, 10th, 11th, and 12th graders in my class. I could use a pie chart to look at the percent of candy I have left. If I have 30 pieces of candy and 20 have been eaten and 10 have not. So there's lots of things you could use a pie chart for. If I want to look at the percent of America, for example, then I would use a pie chart, or a percent of plants that grew, something very general like that. A diagram is simply a labeled picture, and it's usually going to show a relationship between objects or parts. So they're used to communicate data in experiments also, or to give just general information. So for example, if you see a labeled picture of a cell, that's actually called a diagram. So last question here. Kudzu is not a native plant to Mississippi. It was imported to control erosion and rapidly became a nuisance. Which type of graph would best show the percentage of Mississippi that is covered by kudzu? A pie graph, a bar graph, a line graph, or a pictograph? Well, if you're looking at percents, you need to think pie. Because it's a percent of Mississippi, Mississippi can equal up to 100%. Pie is going to be your best option. So anyway, that concludes this set of notes. Please understand that this set of notes is done over multiple days in class, which is why this is a lengthy video. So, don't fret. I guess I should have said this at the beginning, but I can go back and add that in. Anyway, if you have any questions, please communicate with me and let me know, because some of this you will be seeing on your test, and I hope this helps you for my class.